everyone, today I'll be introducing our study on the intersection of modern technology and environmental stewardship. Our primary objective is to understand how we can apply molecular markers and genomic techniques specifically on the purpose of plant conservation. Okay, so how we actually do this? We use two main scientific approaches. First, we use molecular markers. They are scientific DNA sequences with a known location of a chromosome. We use them to identify and differentiate between individuals, population, and species. Second, we use genomic techniques. It involves studying the entire genome, which is a complete set of DNA within an organism.
Now let's talk about the importance of plant conservation. Plant conservation is the essential practice of protecting and restoring plant species to prevent extinction and safeguard our planet's future. As primary producer, plants provide critical ecosystem services by regulating the climate, purifying water, and also supporting global food chains. By protecting plant biodiversity, we maintain the foundational health of our environment and the economic stability of the industry that depend on them.
Type of genomic techniques. First, we have DNA sequencing. DNA sequencing is a method to determine the nucleotide basis (ATGC) in DNA. The second one is genome editing. CRISPR Cas9. It is a direct modification within living cell. The third one is a PCR. PCR means polymerase chain reaction. It is a technique that capable of amplifying specific DNA fragment in vitro. The last one is a microanalysis. It uses a DNA copy to measure a gene expression pattern and to detect mutations.
Type of molecular marker. RFLP
is caused by change in DNA sequence. These changes are detected using a resistant enzyme. RFLP is a codominance marker and use hybridization technique. It was one of the first molecular marker method and it highly specific to certain DNA region.
AFLP
is a dominant marker. It combats a resistant enzyme digestion with a selective PCR amplifications. It requires only a small amount of DNA and doesn't know in a pure sequence informations.
RAPD
is also a dominant marker. It is based on a PCR but the amplified DNA fragments are random. Sequence information is not required and only one prime is used. RAPD produces the high level of a polymorphism and it's simple to perform.
Now we'll talk about importance of molecular mapping in plant genetics. Helps identify key genes that control important plant traits such as yield, disease resistance, and stress tolerance. This makes it easier to understand how these traits are inherited. So detect genetic variation by comparing differences within and between plant populations. Allow scientists to assess the level of genetic diversity in a species. Molecular mapping also guides conservation decisions by identifying unique and threatened plant population, ensuring that valuable genetic resources are protected. Lastly, molecular mapping helps monitor inbreeding and genetic erosions, especially for small population, which is important for preventing genetic problems and ensuring long-term survival.
The title of the journal is The Development of Plant Genome Sequencing Technology and its Conservation and Application in Endangered Gymnosperm. The journal background. Genomic techniques help conservation genetics through enabling high-resolution analysis of genetic diversity, assessing breeding and adaptive potential of the endangered plant species. It also helps in developing effective conservation strategy and ensuring the long-term survival for the endangered plant species. The genomic technique used in this journal is NGS, which is Illumina Solexa, TGS, which is PacBio sequencing. The third one is DBS. Next part is what they do. So the first one is NGS, which is Illumina Solexa. It supports the genomic studies in non-model species where reference genomes are unavailable. This TGS, which is PacBio sequencing. It supports the population genomic and phylogenomic studies to identify genetic variation. Provide the genetic information such as finding the population with low diversity or strong adaptive potential trait. C is DBS. This helps in assessing the genetic diversity and population structure of the endangered species. Also help in detecting gene flow and inbreeding among the fragmented population. The first impact is supporting the conservation planning through the data that show how populations are linked or separated as an important information on gene flow and isolation. Next, it is important to understand the population genetic variation and the potential inbreeding within the endangered species before building the appropriate conservation strategies. Lastly, it enables detailed assessment of genetic diversity, population structure, and gene flow among fragmented or threatened population.
The journal I reviewed is titled Potential of Genomic Approaches in Conservation of Plant Biodiversity in Africa. This study highlights that genomic techniques play a crucial role in plant conservation by providing high resolution information on genetic diversity, population structure, and adaptive potential of plant species. Genomic approaches help researchers identify plant population at risk, design effective conservation strategy, and support the long-term survival of threatened plant species. The main genomic techniques used in this study include next-generation sequencing (NGCs) such as Illumina, HiSeq, and NovaSeq for large-scale genome analysis. The second one is third-generation sequencing (TGS), including PacBio and Oxford Nanopore, which allow long-read and more accurate sequencing. The last one is genotyping by sequencing (GBS) to assess genetic variation within and between plant population. And what they do, first one is next generation sequencing (NGS) provides detailed information on the population genetics of threatened plant species and support genomic studies in non-model plant species even without reference genomes. For third generation sequencing (TGS) support population genomics and phylogenomic studies to identify genetic variation. Also provide data on population with low genetic diversity or important adaptive traits. And genotyping by sequencing (GBS) monitors gene flow and inbreeding in fragment plant populations and helps prioritize population for conservation based on genetic health. The impacts of these genomic techniques include support conservation planning by revealing how plants population are connected or isolated, helps understand genetic variation in breeding rates before designing conservation strategy, and guide restoration effort by identifying plant genotypes best suited to survive in changing environment.
Title of the journal is a molecular marker in plant breeding. Background: plant breeding has traditionally revealed on visible traits, but this can be impacted by environmental factor and might not correctly represent a trait through genetic basis.
Molecular marker provide a DNA-based approach that allows breeder to detect genetic differences directly, making the selection more accurate and efficient.
Marker that are used are RFLP, RAPD, AFLP, SSR, and SNP.
What they do: first, help to analyze genetic diversity by revealing real genetic differences and the relationship between plant lines without being affected by environment factor. Secondly, able to group and manage genetic collections and identify duplicate excisions that improve the efficiency of germplasm conservation program. DNA profiling can be used to identify a variety and hybrid which help to confirm a varietal identity and maintain a genetic purity in a breeding and seed productions. Marker assisted selection (MAS) allowed breeder to choose plant with desirable traits that an earlier stage, even before the traits are visible, which save both time and resource. Impact: first, molecular marker reduce the need to rely on visible traits which are influenced by the environment and make plant selection more accurate. They enable faster, more precise selection, particularly for a complex traits controlled by multiple genes. This marker help manage plant genetic resource more efficiently by providing detailed genetic information. Lastly, support accurate identifications and the development of the improved plant variety, making a breeding program more efficient and dependable. Breeding cycles can be accelerated because selections can take place at the seedling stage, shortening the overall times need to develop, test, and release a new plant variety compared to traditional field methods.
This study is based on a journal article titled by development of DNA markers for identifying the origin of endangered rosewood Dalbergia oliveri in illegal trade. Rosewood is actually the world's most trafficked wild products. Illegal loggers often bypass international bans by mislabeling timber from protected forests as legally sourced. This makes it very difficult for authorities to track and stop. Researchers use molecular markers such as SSR, simple sequence repeat, and SNP, single nucleotide polymorphism. Here is how the process works. Researchers collect leaf or cambium samples from Dalbergia oliveri across its natural range. By analyzing SSR and SNP, they identify region-specific alleles and genetic patterns to construct a comprehensive reference library. This genetic map serves as a vital tool for determining the precise geographic origins of individual timber samples. Researchers also compare the genetic fingerprint of the seized timber against the established reference map by using specialized computer algorithms and assignment testing. By calculating the probability of a match across various geographic populations, these models can pinpoint the timber's origin with high statistical confidence. This measure has two impacts. DNA profiling offers an objective and immutable identification. By utilizing this data, prosecutors can definitively prove in court that timber originated from a protected no-take zone. This provides the forensic certainty required to secure higher conviction rates for international traffickers and effectively dismantle illegal logging networks. Conservationists can also verify seeds that are replanted within the native ecological zones by mapping genetic structure of different regions. This strategic approach can prevent outbreeding depression.
Let's move to challenge, limitations, and future difference. For challenge and limitation, we have four. First is high cost. The cost of high throughput sequencing and bioinformatics is high, which might be limitations when conserving biodiversity developing biodiversity hotspot. Second is technical complexity. The complexity of genomic data, often referred to as a big data, also requires specific technical experience. In bioinformatics, quite often conservations, biologists, ecologists themselves, but do not have the technical knowledge required for the analysis of complex genomic data. The third one is quality of genetic material. In some cases, interacting high-quality DNA from hybrid specimen or from plant found in remote area can be difficult due to degradation or the presence of secondary metabolite that can interfere with sequencing. And lastly is gap between data and action. Just having genomic data is not enough. It does not always ensure that a policy shift will occur. It is often observed that there is a gap between existing lab result and implementing this result for conservation management. For future direction, genomic going past the single reference genomic to scale the complete genetic diversity with species. Second is CRISPR and gene editing. Possible utilization for gene editing to assist threatened flora with adapting to the rapid increase in climate change and developing resistance of emerging diseases. The third one is portable sequencing. This involves applying the use of portable devices such as the one developed by Oxford Nanopore to do genetic surveillance. And lastly is integrations of AI. The application of machine learning to forecast the responses of particular genotype for future stresses. To conclude, the coupling of molecular market with modern genomic analysis has put the field of plants conservation from being responsive to being predictive in nature. The power to offer scientific release on the ability to see past of physical characteristics for declining species and understand the genomic integrity on plant species under the threat of extinctions. By being able to identify conservation units based on the ability to follow the pattern of a gene flow, scientists are able to have the information required to respond accordingly to the habitat restoration projects and the banking seeds for conservation purposes. But for the full potential of this benefit to be realized, the scientific community at large must be able to compete on equal playing field based on resource issues to ensure the development of genomic technology for the scientists in the world's most biologically diverse region by providing, as mentioned by Hollywood 2020. The combination of traditional ecological data and the applications on genomic analysis to reverse the rates of plant species lost in the face of the modern climate change at a rapid rate.